Hey, welcome back to the Classics of Immunology Journal Club. And this week we're going to discuss another classic paper from 1968, that wonderful year where everything turned topsy-turvy in the world. Parisian students rebelled and so forth and so on. So if you like the video, if you like the Journal Club, like it. Don't forget to notify on the little bell. And then go on, also scroll down and go to all of the things that are on my website. And you might like my Twitter feed, for example. And I also have an Instagram site where I have some of my artwork that you might be interested in. Today's paper was published in 1968 in the Journal of Experimental Medicine, which, even though it's called the Journal of Experimental Medicine, it was started in the early part of the 20th century by the Rockefeller University when there weren't too many other journals around. And at that time, in the first half of the century, basically medicine, at least experimental medicine, was really microbiology and immunology, because they were right in the throes of the whole bacteriology discovery of new bugs and causing many of the diseases that the medical doctors had been working with for centuries beforehand. So the Journal of Experimental Medicine should be the Journal of Experimental Immunology. This was one of the classic papers that came from that. And the authors here are interesting: Emil Unaneu and Brigitte Askonas. The title of the paper is "The Persistence of Immunogenicity of Antigen After Uptake by Macrophages." So last week we had a paper from Don Mosier from 1967 in Science, essentially showing that without macrophages, lymphocytes didn't know what to do and they couldn't make any antibodies. And so now this paper, coming a year later, is one of the first experimental approaches to try to find out, so what are these macrophages doing in there? How are they programming the lymphocytes? Because it was clear by then the lymphocytes were the cells that were then differentiating into plasma cells and making large quantities of antibody. That much was pretty solid and had been established. The fact that you needed macrophages was only approachable after Michel and Dutton, also in 1966-67, devised this new assay method to activate lymphocytes, spleen cells, lymph node cells in vitro with sheep red blood cells, and then reading out antibody production in the so-called Niels-Jerne plaque assay. So that's the stage that we're on now. And this was, this paper coming in 68 this is now from England, from the National Institute of Medical Research in Mill Hill in London. So, the word was out. Macrophages are important. We got to figure out what they're doing. And a whole bunch of people were working on this. And not only Don Mosier as a PhD student at the University of Chicago. So to give you a little background on Emil Unaneu, he was quite the man. And he was, I know him well. He was always a little hyperactive, and excitable kind of a person. He was from Cuba, and he graduated from medical school in 1960. And Castro marched into Havana on New Year's Day of 1959, and that was the Cuban Revolution. So that was sort of a hairy time, I would imagine, to be a medical student or any kind of a student at the time. But Unaneu, soon thereafter, he got out and he went to the University of Pittsburgh. And he worked there only briefly for a while. And then I think what happened to him is that his mentor, Frank Dixon, moved to Scripps Institute in La Jolla, California, and Unaneu went with him, or soon thereafter. So he spent some time on the coast, on the Pacific coast in Southern California. And Frank Dixon was into glomerulonephritis. And so Unaneu worked on those projects for a while. And then after a year or two, three, he then emigrated to England, to London, to work with Brigitte Askonas, who was an immunologist at the National Institute of Medical Research in London. this paper is one of the papers that Unaneu and Askonas did together. Now, Askonas is another person, so Unaneu graduated from medical school when he was 26 years old. And so at the time of this paper, in 1968, he was around 33, 34 years old. Askonas was like 10 years older. And her life was disrupted by or changed by World War II, because her parents were Jewish, they were originally Czech, but living in Vienna. And when Hitler invaded the Austria in 1938 or so, they fled, they got out. They had converted to Catholicism, but they got out anyway because, so they got out and they emigrated to Canada. And Brigitte then ultimately went to the university in Montreal and McGill. So when she left Austria, she was only 15 years old, and they went to Canada. She then went to the university, majored in biochemistry, and then moved to England in 1949-50, and went to Cambridge and became a graduate student and got a PhD in biochemistry. And so she got into protein chemistry and so forth. After she got out of her doctoral years, she got her first job was in Mill Hill at the National Institute of Medical Research. And so she was around 29, 30 when she did that, in 1952. She was around 45 years old in 1968. So she was just 10 years older than Unaneu was. And she had gotten interested in immunology. And of course, as we've gone through these classic papers, but in the 30s, 40s, 50s, and so forth, it was much more of an in vivo experimental process to try to understand how the immune system was working. And this paper is sort of a cross between the in vitro and in vivo. In their introduction of this paper, they go through sort of the, what was known. What are the known observations in this business about the question was, what are macrophages doing in order to prompt the lymphocytes to make antibodies? They knew at the time that there were two different kinds of phagocytic cells that would gobble up things and digest them. And so they were phagocytes. There were the macrophages, the big eaters, and there were the microphages, that were the little eaters. There were more microphages than there were macrophages, but be that as it may. And those two names came from Metchnikoff from the end of the 19th century when he was with Louis Pasteur in the Pasteur Institute. So everybody knew that these, because they could see it under the microscope, which is how Metchnikoff first figured this out. You could see them engulfing things, macro particles, and then digesting them essentially. And of course, as I said, Don Mosier showed that macrophages could help lymphocytes make antibodies. So that was known. And it was also known that macrophages seem to persist inside. Antigens seem to persist inside macrophages for quite some time thereafter, but it was sort of qualitative experiments. They hadn't been really dealt with carefully. And the other thing that they knew is that people knew was that macrophages seem to digest and catabolize whatever they ingested. Now, so what these people decided to do was to try to figure out the immunogenicity of macrophages, to try to find out whether or not it was whether the macrophages, after they ingested antigens and so forth, they could how long they could remain antigenic, immunogenic, and help the B cells make antibody. Was this a persistent phenomenon over a long time period, or was this just a little short thing that happened? So that's what the paper was all about. And they decided for the antigen that they were going to work with, they picked a huge protein, hemocyanin from a squid. The hemocyanin is a molecule, it's a protein. in invertebrates that is able to interact with oxygen and supply oxygen to the body. And I'm familiar with keyhole limpet hemocyan, and a limpet was a sort of a barnacle that had a hole in the middle of it. That hemocyan, and I know was 7 million was the molecular weight. I don't know what I think the hemocyan and that the squids had was a similar, very large molecule. So it was a good antigen because it had all kinds of places, since it was such a big molecule, it had all kinds of places on it where antibodies could attach to it and so forth. The assays that they used in the materials and methods of this paper, they needed an assay for antibodies once the B cells made the antibodies. And so what they did was they attached some of the hemocyan and onto sheep red blood cells. And then they could just look for agglutination from the serum of an immunized animal to see if it would clump up the red blood cells. So that was a hemagglutination assay. And for macrophages, the source of macrophages, everything was in the mouse, and it's probably going to be for the next 100 years, everything's going to be in the mouse in immunology. The other people had already shown that if you inoculate it into the peritoneal cavity of a mouse, so a gamish of digested proteins, proteose peptone, which you could buy at the store from some of these biological supply houses. And that was just basically food for the macrophages. It would draw in the macrophages into the peritoneal cavity, and they'd start gobbling up all these partially digested proteins, essentially is what that is. So if you injected this stuff into the peritoneal cavity, you waited 3 days, you could get 80 to 90% of the cells that would come into the peritoneal cavity, and a whole bunch of them would come in there, so you could get lots of cells for your experiments. But in addition, they were pretty, not bad because they're 80 to 90% macrophages after 3 days. If you wanted polymorphonuclear leukocytes or microphages, you only had to wait 30 to 45 minutes. The first cells that would go into there would be the PMNs or the microphages. So that was a good thing. You could compare then microphages to macrophages. Particularly, they were interested in the immunogenicity, and they found, well, I'm getting ahead of myself. So then you could take these cells out after 3 days for the macrophages and put them in tissue culture, Eagles and MEM again, and they use normal mouse serum at 5%. You can put them into culture flasks, and they wanted to keep the macrophages, as I think I said before, they'd settle down on the flask, on the plastic and or glass, and they would attach. So then you could wash off the lymphocytes if you wanted to. But if you wanted to retrieve the macrophages, as I said before in one of the videos, you had to use a rubber policeman to scrape them off. So these people put a magnetic stirrer in their flask and put it on top of a magnetic stirrer, kept everything into solution, and the macrophages would stay up in the solution. They wouldn't attach to the glass, which is what they wanted. They found that they could culture these cells up to 2 days, 48 hours, and everything was pretty good. And they didn't lose any cells and so forth. But after 48 hours, after 2 days, things started to drop off, cells started to die, and it was clear that you couldn't really extend anything beyond that. And so they got that far, and now they needed an assay for immunogenicity of the macrophages. And this gets a little bit complicated, and they refer to a whole other paper that also came out in 1968, this one in Immunology, the British journal, where they established the experimental procedures in order to test whether the macrophages could continue to be immunogenic or antigenic. And so what they found is that if they just inoculated the hemocyanin into mice at pretty low dose, they didn't really get a primary immune response that they could measure. But if they inoculated macrophages that had already ingested the antigen, then it would prime those mice, the macrophages would, so that they could come back, 3 weeks later and inject the mice with soluble hemocyanin. And now they'd get a secondary immune response that was very easy to measure in the serum. They could take the serum, they could do their hemagglutination assay, and now they had a system that they could work with. So that's the story. Those are the methods that they used. And so they're doing in vitro cultures and they're doing in vivo experiments to try to figure out how the immune system deals with all this business and focusing on the macrophages. And so in the results section of this paper, what they wanted to find out first, how much of the hemocyanin, in this huge molecule, was phagocytosed over time and degraded. And so the way they could do that was they labeled their hemocyanin with a radioisotope, I-131, iodine-131, that they could then, so that's a tracer, and they could then take cells and whatever they wanted to, they could measure that in a radioactive counter, gets counts per minute as a readout, and that was quantitative so that they could ascertain how much of the radiolabeled remained with the cells over time. And in addition to that, what happens to the radiolabeled hemocyanin is that when it gets digested and catabolized by the cell, then the radioisotope tag is released outside of the cell and it's not reutilized. It's also no longer associated with large molecules, and so it's not precipitable by trichloroacetic acid. So the TCA. And TCA then could be used as a method to separate digested radioisotope from non-digested radioisotope. On the basis of that, they could find out how rapidly the macrophages would chew up and digest this stuff. And they found that after 2 or 3 hours in culture, the macrophages would catabolize about 80% of the antigen. And most of it, over 90% of the radioisotope of the radiolabel was not TCA precipitable. That is, it was not associated with large molecules anymore. And they found that only 10% of the initial radioactivity that the cells gobbled up was retained after 5 hours. And so that was interesting because, if the antigen is being chewed up in tiny little pieces so rapidly, how can it be immunogenic? That was the major question, essentially. And they did autoradiographs, which they could make smears of the macrophages and then expose them to a radiographic plate, like an X-ray. And then they could count the number of grains of radioisotope in the different cells under the microscope and so forth. And they found that when they would incubate the radioisotope tagged antigen with the macrophages at time 0, 75% of the cells were positive. So, the vast majority of cells. And after 4 hours, 69% of the radiolabel was still associated with cells. So that gave them a little bit of figuring out what they could get away with in the short term of this whole thing. But they needed to know how long these macrophages might be immunogenic. And so they used their immunogenicity assay of first taking the macrophages after they've been cultured at different time intervals and so forth, injecting the mice and then coming back later with the soluble antigen and measuring the secondary immune response in the serum. And they found that the immunogenicity, and this is the critical part of the paper, was persisted for at least 2 weeks. So that was a very long time period. And they were surprised at that. I don't think they knew what they were expecting, but they expected it was that long. And that was as long as they did it for. It was 2 weeks. They didn't do it longer than that. But that was it. That was the paper. And so they go into the discussion then. And in the very first sentence in the discussion, they say that macrophage immunogenicity persisted for a long, long time with only a very small amount of antigen remaining in the macrophage. So that was sort of, how can just a tiny little bit of the antigen be antigenic for such a long, long time period? The other thing that they wondered about was, well, where is this antigen? Because it seemed to have escaped the digestive catabolic process of the macrophage. Most of the antigen got chewed up, but the immunogenic part of the antigen that was still in those macrophages seemed to be protected in some way. How could that happen? Why didn't the cell eat it all up 100% rather than only 90%? So they wondered where it was in the cell that it was protected. They said that they couldn't discern that from looking at the cells under the light microscope and the autoradiographs and so forth. But they say in the text of the paper that they've done electron microscopy. This is in the discussion, though. They didn't give any results, and that they were going to publish that in the next articles, which I've read, and they found it in the membrane. So I'm cheating here a little bit and going a little bit forward for you. So then the question was that they talked about in their discussion was, okay, so only a little bit remains in the macrophage, and it seems to be protected from the digestive process and so forth. Well, how do the macrophages interact with the lymphocytes? What happens? The lymphocytes, do they touch one another, or is that important? And how do they do that? It just wasn't clear. They knew that there was a lot of stuff out there at the time that said that perhaps RNA had something to do with immunogenicity. And so they treated their whole things with enzymes that would digest RNA, RNases, and they found that didn't have any effect on the immunogenicity. So that they thought that RNA was a red herring and wasn't involved, and that was true. But they wondered whether or not there in some way that the antigen that persisted in the macrophages was somehow changed. It was processed, which is the word that that came to be used in the whole business. And then finally, they wondered about a lot of things. So they're setting their course up for the next 10 years. They've got all kinds of experiments that they can do. They wondered whether or not, in addition to this antigen processing and so-called presentation, which became a whole new area in cellular immunology, they wondered whether or not the macrophages somehow influenced the lymphocytes, prepared them in some way. And of course, we know the answer to that as well. And that is, yes, they did. They made cytokines. And they made cytokines that really influenced the lymphocytes. And we'll get into that soon, actually, very soon after that. So the macrophages are an interesting cell. It's doing all kinds of things in the immune response. And, the focus for most of the immunologists, and as I said before, for the whole first half of the century was only about antibody production. But we're getting into the 60s, they were for the first time finding out how the cells made the antibodies and also the immunochemistry of that had been going on forever and ever as to what the antibody molecules actually were, what they looked like. And that happened and finally came to fruition around 1960 as well. So that's it for this paper and for this week in the classics. Don't forget to, if you really like this video, let us know. Like it, click on the bell, go down, scroll down, and get involved in all the other things that you can in this wonderful area and this wonderful world of immunology. Thanks.